Thank you. So I'm Pavol Bokes, for those of you who do not know me. I'm from the Department of Applied Mathematics and Statistics. And I will tell you something about feedback on dilution in stochastic gene expression, a comparative study of single-cell and population frameworks. There are two important names that I should mention. First is Irina Zabaikina, who worked on this as part of her PhD project. And the other one is Abhi Singh from Delaware, who initiated this project. And I'm the middleman. Yeah. Okay. Stochastic gene expression. So it's all going to be about stochastic gene expression. So we pulled some figures from literature, which give a basic idea what stochastic gene expression is about. When you look at this, so here you have time, that's the horizontal axis, and the vertical axis is the number of protein. So genes produce proteins, and we are interested in the number of proteins. And as you can see, the production of proteins is not continuous deterministic, but occurs randomly, right? So it occurs in these randomly timed and randomly sized bursts of rapid production, and then it stays constant again, and then you have a burst of production and again stays constant. So the protein is called beta galactosidase, I think. It doesn't matter, any kind of protein you can think of, right? So this stochastic gene expression looks like this. And the other figure is a histogram, which, well, somehow don't know what's happening here. Yeah, this is the thing. Yeah, so this histogram that shows you the histogram for the sizes of these bursts. So as I said, each burst is randomly timed and randomly sized, and the sizes are random, and the distribution is like it shows on the figure below. So on the horizontal axis, you have the size of the burst in number of molecules, and in the vertical axis, you have how many times you have seen such a burst of such size. And the exponential distribution fits it quite nicely, as you can see. So this is exponential distribution, the fit. It's a simple exponential curve. And it actually makes sense because exponential distribution is a memoryless distribution. It has some physical meaning behind it, why it should be memoryless. Okay, so this is taken from one of quite influential papers from around 20 years ago, which were looking at gene expression at single molecule level. Okay, so this is one piece of information that we are working with. The other piece of information that we will be working with is that the number of proteins can have an impact, and we will assume that the number of proteins in the cell has an impact on the growth rate of the cell. So here is a specific example of it. Again, it's pulled from literature, where you have on the horizontal axis, you have the amount of protein. Coincidentally, it's the same protein, which now I can read because it's full galactosidase, right? So that's the protein. And the vertical axis is the growth rate. And in this specific example, the number of protein negatively affects the growth rate of the cell, right? So the cells grow and then they divide. And if you have a lot of protein, the growth can be slower. One reason mentioned here is that you can have yeah, it's sort of diverts resources from other processes. So that's one mechanism how the number of proteins can affect growth rate. So we wanted to combine these two things. First is the stochastic burst expression, and the second one is the dependence on growth rate. Okay, so here's how we want to do it in terms of modeling. So we are thinking about a single cell, and here's our model for how single cell grows. Right? So we start with a small cell on the top left, with a small volume. Then it grows. It reaches a critical volume, and when it reaches the critical volume, it divides into two daughter cells. So the daughter cells will have the volume which the mother cell had at the beginning of the cell cycle. It's called the cell cycle. And we are just looking at the single cell in our first model, so that means that we forget about one of the daughters and we follow only one of the daughters we follow, and yeah, that behaves exactly as its mother. Okay, and for this one cell we are tracking two variables. First is volume, so that's volume of the cells, which increases. And the second one is the concentration of protein. Concentration, not the amount of protein, but concentration. So for this concentration, we have this bursty model that the concentration is increased in random bursts of exponential size, and the alpha is the rate with which those bursts occur. So they occur like a Poisson process randomly in time, and the size of the burst is capital B, and that's taken from the exponential distribution as suggested by experimental evidence. We also have a dilution there, right, because it's concentration. So as the cell grows, the volume increases, and as the growth increases, as the volume increases, the concentration gets diluted, right, by this growth. So we have dilution of concentration, and it is obvious that there's a coupling between the growth, the rate of growth, and the rate of dilution, right, because one causes the other. And as I said, we also have the effect of protein number, the protein concentration on the rate of growth. So there are some effects which we have here. Essentially here we have two differential equations which say that the relative change in growth, logarithmic change in growth in volume, and logarithmic change in concentration, they are the same. And we model it by this kind of decreasing function. Gamma max is the maximal growth rate, and then it decreases as you have more protein concentration, you have more cost to the growth. So that's the denominator, which is here 1 plus k times x. So k is the proportionality factor. So it tells you how much the protein reduces the growth, and and at the same time, so reduces the growth means slows down its decay. So the protein slows down its decay, its dilution, and so when you have a lot of it, you have slower dilution. So which means that we have something which we call in biology positive feedback, right? If there's a lot of it, then you have a tendency to stay with a lot of it, and if you have little of it, then so this is what we call positive feedback. In biology, it's not positive feedback in student. How do we call it? Student anketa, right? It's not this one. Right? It's biological positive feedback. And the left-hand side shows you, that's how concentration decays for different values of k, which is the feedback strength. We call it feedback strength because the higher the k, the stronger the feedback gets via dilution, feedback via dilution. Right, so for different k's, how you have different decay patterns and how you have different growth patterns, that's the figure downstairs. All right, so let me show some simulations of this model. So we have two versions. So the first one is k equals to 0. There you have no effect of protein on growth. That means no feedback. And the right-hand side is we have k positive. That means we have feedback. The first graph, both left and right, is protein concentration as function of time. And the second graph is volume of a cell as function of time. So this is a trajectory of something that we call PD-MPD, piecewise deterministic Markov process. Because it's piecewise deterministic, you have only these bursts, single events where something stochastic happens, and then between those bursts it behaves deterministically. So that's the PDMP, piecewise deterministic Markov process. And it's Markov because kind of all processes we know enough about our Markov. So there is one difference between the left-hand side and the right-hand side, which is kind of interesting mathematically. Which is interesting mathematically. So when you don't have this feedback, then volume increases periodically, right? It increases kind of, this is exponential growth of the volume, and then it reaches critical volume when its cells divides and goes back here. So it's a periodic function. And when you have this deterministic periodic function in a stochastic process, then it's kind of obvious that this distribution of the stochastic process doesn't converge to a stationary distribution. The process will not be stationary. The distribution we denoted by fxvt. So that's the probability density function of variables x and v at time t. And it doesn't converge as t goes to infinity as time progresses, right? Because kind of this periodic motion of the volume keeps the information about the initial condition, and you can't have a convergence. Whereas where you have positive feedback, which means this thing that, let's say if you have a big burst here in protein concentration, and what happens is that you slow down the growth, right? So the growth slows down, and then it's no longer periodic. And when you don't have this periodicity, then it's reasonably to assume that you have a distribution, steady-state distribution as time goes to infinity. Of course, here should be v instead of t, which is obvious. So how do we model this kind of processes? This is a general formula for the distribution of a continuous state-space process, fx. X is now a vector. In our case, it's xv, concentration and volume, at time t. And so the PDF, the probability density function, satisfies this evolution equation. Evolution, not biological evolution, but evolution like in terms of evolution of time. So the temporal derivative is this differential operator, differential integral operator. And you can see there are three parts to it. So the first one is drift. So that's continuous drift, right? That's deterministic motion. Then you can have something like diffusion, which is the second derivative operator. And then you can have jumped, which are discontinuous jumps in time. So in a general process with continuous state space, like we have concentration and volume, you can write this kind of a partial differential equation, partial integral differential equation, which is called Chapman-Kolmogorov equation. Assuming some moment conditions or something. But Yeah, and also you can have some boundary condition and initial conditions which complement this and specify this problem. In our case, we just have the drift term, right? So this is our model. We have the drift term and the jump term. We don't have any diffusion term. So this is the drift term, and you can see the rates of dilution and the rates of cell growth there. And we have this jump term, and you can see the exponential distribution with mean beta there. Beta is the mean size of the burst. So we have this equation. And we also have a boundary condition which tells you what happens as the cell reaches the maximal possible volume, and then it goes back to the small volume. It's some kind of flux preservation condition. Right, and steady state, you can look for the steady state distribution. As I said, in general, when you have this feedback, you expect a convergence to a steady state distribution, and that's what we are after. And nice thing is that you can solve it by Fourier method. That means separation of variables. I don't know what's happening now. Control L. Yeah, so you can solve it by Fourier method, separation of variables, and you can get a nice analytical solution. I mean, me, not you, maybe you, not me, but Irina did, yeah, got it, yeah. so Irina got a nice analytical solution with some condition that between production and growth rate, which is somewhat technical, but we have an explicit solution. Okay, so this is single cell results. Now there's a slide about how these single cell measurements are made, and the bottom line is that they are kind of difficult. And instead you would like to have easier measurements, but easier measurements require cell lysis or cell death. So easier measurements is that you grow the whole population, and then at a certain time you kill the population and count the number of proteins in each cell. In that case, you are not looking for a single cell process, but you are thinking about a population process and population distribution. So we looked at how to do populations instead of single cell. So here's the slides that I began with, but it's a cell population model. So you have the mother cell that divides into two daughter cells, and daughter cells divides into further daughter cells. And now we track the concentration of proteins and the volume for each of these cells that we have in the population. Okay, the rest of the assumptions are the same, like the division rate, protein dependent, bursts, and so on. Here are some simulations. We also call it branching process. Branching because the cells branch into their progeny. And again, there are two versions of it. So the first one will be positive feedback, where you have feedback k greater than 0, and there's no feedback. Okay, so how it looks pretty much the same as with single cells, but when you divide, you track both daughter cells. So you now have two trajectories, right? And then again, one of the daughters divides and you have three trajectories and so on. Mathematically, you can think of this as a measure-valued Markov process. So that's the way to represent this mathematically, whereas at each time you have an empirical measure of concentration in the concentration time volume space, right? So each cell is represented by a delta function, and you add up the delta functions, and then you have an empirical measure. So it's a measure-valued Markov process, if you want. And yeah, and again, the same story holds about the periodicity. So when you have no feedback, you have periodic process, and you can't expect convergence of this process, measure value process, or whatever. And when you have feedback, then you get rid of this periodicity, and you will have convergence. Okay, this is population balance equation. I'll just scroll it to our model. So this is our model. So H is the population density, right? So it tells you how many cells you have with concentration X and volume V at time T. And the funny thing is that the equation looks exactly the same. This is the same equation, but instead of F, I have H. The only thing that is different is the boundary condition. And it's kind of logical because boundary condition tells you what happens as the cell reaches the top volume and then it divides, right? So in this boundary condition is the division. And instead of 1/2, we have 1/4 here. So it's both the conservation of fluxes, but it's not quite conservation because when you move from volume 2V star to volume V star, which is the critical volume, then you double the number of cells, and that's in the boundary condition. Again, nice thing about this model is that it is solvable, so you can again use the Fourier method, and after a bit of algebra, you get a solution, which is not yet here, but the lambda is here. So lambda is this part of the Fourier. It's the rate with which the Fourier mode grows, right? So it's the large time growth of the population. Yeah, so we have a formula for lambda. So that's the Malthusian parameter for the for the cell population. And this is the graph which kind of checks it against simulations. So the horizontal line is time, and the vertical line is population size log scaled. That means that the straight line corresponds to exponential growth. And the straight lines are the theoretical predictions with lambda as we calculated them, and the stochastic lines are the results of simulations of the branching process of the measure valued Markov process. And again, we have some condition for the existence of the distribution, which I won't go into detail about. And yeah, this is the exact distribution, and here is a comparison against stochastic simulations. So we have these are histograms, how many cells we have with what kind of concentration, how many cells we have with what kind of volume, and we counted it using simulations, right? We did the simulation of the branching process Irina did. We got 14,000 cells at the end of the simulations, and we counted how many of them have which concentration of protein, which volume, and these are the histograms, and it fits with the theoretical prediction. Yeah. So here are some summary. So this is we looked at the theoretical formula, and we did some analysis of it. So essentially, when there's no feedback, okay, so this is like the rows are no feedback and feedback. So the upper row is no feedback, and the bottom row is feedback. And then the columns are concentration and volume. These are distributions, right? Large time distributions. And we look at population distributions and single cell distribution for each combination of these two things. So when you don't have feedback, population and single cell is the same distribution, gamma distribution, which is well known from earlier studies. This is the, on the right, there's the distribution of volume. So here you have a different distribution in the population case and different distribution in single cell case. Essentially, when you have population when you look at the population case, then the population has more small cells. Yeah? It has a tendency to prefer smaller volumes because those cells grow. And feedback has no effect on the volume distribution, so that's this. But feedback has effect on the concentration distribution, and you get different distributions for population and single cell. So the bottom line is that the population distribution is light-tailed compared to the single cell distribution, which can be important if, let's say, cells above certain cells which have protein above certain threshold, which would be these cells here. Let's say they are important in some sense. For example, they are drug-tolerant bacteria or cancer cells, which is something that we are working on with Abhi things in other context. Okay, I think I should rush. Thank you for your attention.